Hello! In today's video, we shall learn in detail about Riccia. By the end of this video, you will have a clear understanding about its morphology, anatomy, different types of reproduction, structure and development of Antheridium and Archegonium, Sporophyte, and the life cycle of the plant. Let us begin with the morphology of Riccia. Riccia is a genus of liverworts belonging to division Bryophyta in the order Marchantiales. They prefer damp rocks, moist soil, shady habitats, though many species like Riccia fluitans are aquatic. These plants are small, simple and thalloid, that is, not differentiated into root, stem and leaf. The thallus may be ribbon-shaped or strap-shaped and usually forms rosettes. The thallus is small, green, flat and grows prostrate on the ground, showing distinct dichotomous branching. Each branch has a broad anterior region and a narrow posterior region. It is dorsi-ventrally differentiated. Its upper dorsal surface is green due to the presence of chlorophyll and helps in photosynthesis. It has a mid-dorsal longitudinal groove on each branch. Air pores on the dorsal surface leads to air chambers. The lower ventral surface has a mid-ventral ridge bearing multicellular scales which are one cell thick, present in one or two rows. They may be violet in color due to the presence of anthocyanin pigment. They are protective in function as they project forward to cover the growing apex of the thallus. Scales help to retain moisture and safeguard it from drought and mechanical injury. Ventral surface also has numerous unicellular hair-like structures called rhizoids. They are of two types. One type is called smooth-walled rhizoids, which are simple tubular structures with smooth inner and outer thin walls. And the other type is the tuberculated rhizoids with internal peg-like projections inside the lumen, which assist in water and mineral conduction and also help in increasing the surface area for absorption. Rhizoids also serve as organ of attachment to the substratum, thus helping in anchorage. At the anterior tip of each branch is a small notch or depression. Inside the notch lies a single apical cell or a group of actively dividing cells. These cells divide and produce new tissues which are responsible for growth and branching of the thallus. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now we shall study the internal structure of Riccia. The vertical section of the thallus of Riccia shows two distinct regions, upper photosynthetic region or assimilatory region and lower storage region. Upper photosynthetic region is present on the dorsal side of the thallus. It consists of vertical column of green chlorenchymatous cells arranged in linear rows. They function as pseudo-mesophyll and are responsible for photosynthesis. These cells are arranged loosely with air spaces or air canals or air chambers between them that communicate with the external environment and facilitate gas exchange. The uppermost cells of this region are colorless as they lack chloroplast and form a discontinuous layer which is known as pseudoepidermis. There are small air pores in the upper pseudoepidermis that leads into the air canals and help in gas exchange. Lower storage region is present on the lower ventral portion of the thallus. It is the lower, colorless, vertical zone of the thallus beneath the photosynthetic zone. It consists of closely packed, undifferentiated parenchymatous cells without intercellular spaces. It contains starch but no chloroplast and acts as the primary food storage house for the thallus. It also gives mechanical support. The lowermost cells of this region are small in size and form a continuous single-layered lower epidermis. Rhizoids and scales originate from ventral epidermal cells. Vascular tissue is completely absent in Riccia. Next is the vegetative reproduction in Riccia. Fragmentation. It is the most common method of vegetative reproduction. The cells in the older portion of the thallus naturally age, die, and decay over a period of time. When the death and decay reaches dichotomy, the two lobes become completely separated from each other. Each isolated lobe undergoes apical cell division, grows independently, and develops into a new thallus. Adventitious branches. Some species like Riccia fluitans develop special adventitious branches on the ventral surface of the thallus in the mid-rib region. Due to injury or aging, these branches get detached from the parent thallus and develop into new independent thalli. Persistent apices. During extreme drought or harsh winters, the entire thallus dries up and dies except the growing apices. These apices grow downward into the soil, remain dormant by becoming thick and encircling themselves with a protective covering. With the return of favorable season, these persistent apices resume growth and give rise to new thallus. Tuber formation. In some species like Riccia bulbifera, at the end of the growing season, the apical region of the thallus lobe is thickened to form specialized underground storage organs called tubers. They store a large amount of starch and nutrients. With the advent of unfavorable conditions, these tubers remain buried and dormant in the soil while the plant perishes. With the onset of suitable conditions, the tubers germinate to give rise to new thalli. Gemmy formation. In some species, small, multicellular, vegetative reproductive bodies called gemmy are formed. They detach from the parent and grow into new thalli. Regeneration. Riccia cells possess a high capacity for regeneration. Any living part of its gametophytic thallus can redifferentiate, divide rapidly, and grow into an entirely new thallus. Next, let us understand sexual reproduction in Riccia. Riccia plant is a gametophyte. Majority of Riccia species are monoecious or homothallic. Example, Riccia crystallina. Few are dioecious. Example, Riccia gangetica. In Riccia, the sex organs are embedded in the median groove on the upper side of the thallus and develop in acropetal manner. Next is the structure and development of Antheridium. The Antheridium is the male reproductive structure. It is pear-shaped or club-shaped and is seen on a short, few-celled stalk. It is present inside the antheridial chamber with an opening called ostiole. A superficial cell that lies on the dorsal surface of the thallus enlarges and functions as the antheridial initial. It divides transversely to form upper or outer cell and lower or basal cell. The upper cell develops into primary antheridial cell and the lower one develops into primary stalk cell, which later forms the stalk. The primary antheridial cell divides, forming outer sterile jacket initials and central fertile androgonial cells. The outer cells divide to form a single layered protective jacket around the antheridium. The androgonial cells divide to form numerous androcyte mother cells. Each androcyte mother cell has a denser cytoplasm and a larger nucleus and divides to form androcytes. Each androcyte develops into a small, slender, spirally coiled, biflagellate anthrozoid. Next is the structure and development of Archegonium. The Archegonium is a flower-shaped multicellular structure. It consists of an elongated, narrow, slender neck and a solen basal portion, a venter embedded in the thallus. The neck has a vertical row of 4 to 6 neck canal cells surrounded by a single layer of sterile cells forming a protective jacket. The tip of the neck is capped by 4 specialized large cover cells that protect the interior canal. The venter is surrounded by a protective, single-layered sterile jacket. The venter cavity consists of one small ventral canal cell and a large egg cell that acts as the female gamete. A superficial cell that lies on the dorsal surface of the thallus enlarges and functions as the archegonial initial. The archegonial initial divides to form an upper cell and a basal cell. The upper cell functions as the archegonial mother cell. It divides to form 3 peripheral initials that give rise to jacket initials, which on further division form the neck and the venter and surround a primary axial cell. The primary axial cell divides to form upper primary cover cell, which gives rise to 4 cover cells and a lower large central cell that forms upper primary neck canal cell, resulting in neck canal cells and a lower ventral cell. The ventral cell forms a small upper ventral canal cell and a large lower egg cell. Next is fertilization. Moisture is an essential factor for the release of anthrozoids. Water enters the antheridial chamber through the ostiole. The cells absorb water and disintegrate. A pore is formed through which the male gametes come out and swim freely in water to reach the archegonia. When the Archegonium matures, the neck canal cells and the ventral canal cells dissolve to form a mucilaginous mass which imbibes water and swells. A pressure is created which forces the cover cells to open, making a passage for the male gamete to reach the Archegonium. Fertilization takes place in the presence of water. The mucilage contains several chemical substances that attract the male gametes. A number of male gametes enter the venter, but only one gamete reaches the egg. Fusion between male and female gamete occurs, resulting in the formation of a diploid zygote. The zygote secretes a thick cellulose wall around itself and becomes the oospore. Next, let us study the development of sporophyte. The oospore gives rise to the sporophyte, which reproduces by spores. The sporophyte is a simple spherical body called the capsule. It lacks foot and seta. The oospore develops within the venter of the Archegonium. With repeated division, it forms a mass of undifferentiated cell known as the embryo. The venter grows and forms a two-cell-layered thick wall known as the calyptra over the developing embryo. The young embryo differentiates to form the archisporium. The archisporial cells develop into the spore mother cells. Each spore mother cell undergoes reduction division and forms a tetrad of spores which are protected by a thick coat. The calyptra and the wall of the capsule rupture and the spores are liberated. The spores germinate by forming a short germ tube which develops into a new gametophytic Riccia thallus. Finally, let us look into the life cycle of Riccia. The life cycle of Riccia shows two different generations: the haploid gametophytic generation, which is dominant and independent, and the diploid sporophytic generation, which is dependent on the gametophyte. The gametophytic plant body bears Antheridium and Archegonium, both of which produce haploid gametes. The fusion of these gametes results in a diploid zygote, which marks the end of the gametophytic phase and the beginning of the sporophytic phase. Thus, there is a regular alternation of generation in Riccia. So today, we have learnt in detail about Riccia. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comment section below. I'll see you there. Goodbye. All the best.